So we're going to continue our lesson in the digestive system, and we are going to start talking about those accessory organs. And so these are going to be the organs that are not coming in direct contact with the food. Digestion does begin in the mouth, and that food substance is going to be physically mechanically broken down by the grinding actions of the teeth. And then we're also going to start having some chemical digestion of carbohydrates by amylase that's released by those salivary glands. And then that roundup food and mix of amylase plus lingual ligase and all those other enzymes, that mix is called a bolus. So then that bolus gets pushed to the back of the pharynx by the tongue. And so that region of the pharynx is referred to as the oropharynx. And then we are going to have the swallowing. So the epiglottis will close, and that will direct the food down the esophagus instead of the trachea. And we're going to have peristalsis contractions move and direct the food into the stomach. So the lower esophageal sphincter will open, and that bolus will enter into the stomach. Now we're going to have mechanical breakdown of the food by the contraction of our three layers, the longitudinal layer, that's the superficial most layer, and then the circular layer, that's the middle, and then the oblique layer, that is the innermost muscular layer. They all contract in different directions, and that is going to really mechanically break down this food. And then in the stomach, we're also going to have the activation of our lipases, like lingual lipase. There will be secretion of pepsinogen that gets converted to pepsin and by the hydrochloric acid. And so we're going to have chemical breakdown of lipids and proteins begin in the stomach. And so that gastric secretions mixed with the bolus is now referred to as chyme. And that chyme is going to get pushed into the duodenum or duodenum, however you want to pronounce it. So that chyme is very acidic. And in the duodenum, there is going to be the secretion of bile and sodium bicarbonate from the pancreas. And so these secretions are important in the digestive process throughout the small intestines. And without that extra secretions, food won't get further broken down, and the acidity of the chyme would eat away at that intestinal wall. So the first organ we're going to talk about when it comes to accessory organs that will secrete a substance into the alimentary canal to aid in digestion, but does not contact the food itself, is the liver. The liver is definitely the largest internal organ, and it has 4 lobes. It weighs about 3 pounds, and it does have a lot of different functions, and we've talked about it in multiple systems. For example, we talked about the liver in hematology with things like fibrinogen and albumin, and with things like hemoglobin. Well, we also talked about it in relationship with glucagon and insulin. But in the digestive system, the function is to secrete bile. And so the bile will help break down lipids or fats. And the liver has 4 lobes. The 2 major lobes that most people are familiar with are the right lobe and the left lobe. The right lobe being the largest. And then it does have 2 minor lobes that are called the quadrate and the caudate. So if we look at the inferior portion or inferior view of the liver, you can note the right and left lobes, and you can also note the inferior vena cava as well as the organ, that gallbladder there. And so the quadrate lobe is this, I always thought like it's quadrilateral, and it's next to the gallbladder. Where the caudate, if you remember that caudal means towards the back, the caudate lobe is the lobe that's towards the back portion of the liver. The liver also has a ligament that will separate the left lobe from the right lobe, and that's called the falciform ligament. And then if you think about where the liver is located, it's suspended from the diaphragm. And so there is a sheet of mesentery that will suspend the liver from the diaphragm, and that's called the coronary ligament. The round ligament is also referred to as the ligamentum teres, but I call it the round ligament. This is formed after birth. It is a remnant of the umbilical vein. And so once the baby is born, the umbilical vein is no longer necessary. It used to carry the blood from the umbilical cord to the liver of the fetus. And so that's not needed anymore. So it kind of just goes away, and it forms this ligament that we call the round ligament. When we look at the anatomy of the liver, the right lobe is going to be bigger. And we can note the right liver, right lobe of the liver, the left lobe. And then here we have that falciform ligament. And you can see hanging right off of that is the round ligament, which is that residual of the umbilical vein. And here we have that falciform ligament there, and you can see the round ligament also highlighted. Here is what the liver looks like in a cadaver, and you can see that ligament here separating the right and left lobes, and then the coronary ligament would be here that is suspending the liver from the diaphragm. So we're going to see this prefix porta a lot when we're talking about the liver, and porta means door. And so porta hepatis is this irregular door or opening between the quadrate lobe and the caudate lobe of the liver. And so what it is it's this point of entry for the hepatic portal vein and the hepatic artery. And then it's going to be a point of exit for bile. And these are going to travel through the lesser omentum. And if we look here on that underside inferior view of the liver, we're going to see the gallbladder. And there'll be a little depression within the liver that the gallbladder kind of sits in. And that depression is on that inferior surface between the right lobe and the quadrate lobe. And then here on the top, we have what's called the bare area on the surface. That is where the coronary ligament connects to suspend the liver from the diaphragm. Here we have the liver and the gallbladder highlighted, and you can see the location of the gallbladder in relation to the liver. You can see that it is located underneath the liver, and that's the head of the gallbladder kind of sticking out there. When we zoom in and look at the histology and microscopic anatomy of the liver, the first thing you're going to notice are these little like octagon or hexagons shapes. They're these little tiny cylinders, but they're not round. They have sides to them. And those fill the interior of the liver. They are called hepatic lobules. They're about 2 millimeters in length and 1 millimeter in diameter. And in the very, very center of them, there will be a little circle that is the central vein that passes down the core of the hepatic lobule. And then we're going to see that there are these sinusoids. So from the central vein, you're going to see these kind of like projections spread out, and they are blood-filled channels that fill the spaces, and they are lined by this epithelium that's going to separate the liver cells from the blood cells. And the liver cells we refer to as hepatocytes. They are a cuboidal cell, so they function for secretion and absorption of substances, and they will surround that central vein, and then they kind of like radiate out into these sheets. So any blood that is leaving the stomach or the intestines doesn't get to just enter into the systemic blood system, right? Like that would be very unsafe because who knows if there's a bacteria that can get into the blood supply. So we are going to send it through a filter first, and that filter is the liver. So all blood that leaves the stomach and intestines will pass through the liver, and it passes through the liver through this portal vein. And that portal vein will deliver that blood from the stomach and intestines, and it will go past these hepatocytes and into the central vein. But if you look between the hepatocyte and the capillary, there is this gap. And because of these big gaps, plasma can seep in between the hepatocyte and the blood vessel. And then these hepatocytes also have this little brush border of microvilli that will project into that space. So blood from the stomach and the intestines will be filtered through this sinusoid, and it's going to come from that portal vein. The portal vein will send the blood through this space, and when it goes through that space, it's going to interact with some cells like Kupfner cells. So Kupfner cells are macrophages that remove bacteria and debris from the blood. So the blood will be detoxified, and hopefully any harmful substance that's in the blood can be removed before it can enter into the venous system and be returned to the heart. So this slide is just what I said verbally on the other slide, just written out for you, so that way you can have it printed in your notes or for reference. In this image, you can see the hepatocytes that are lining the sinusoids, and those are coming into direct contact with that blood. They are secreting substances into the blood, and they are also removing substances out of the blood. And so if we look at this image over here to the left and we follow the blue, that's representing blood coming from the stomach and the intestines. So this is that portal vein, and the blood drains down through these sinusoids, and then as it does, it's going to have contact with these hepatocytes. And then it's going to go into the central vein. So all of these sinusoids drain into the central vein here. You can notice that little macrophage. Now let's look at the red part right here. This is a branch of the hepatic artery, and it's providing the oxygen and nutrients that the liver cells need. So this is not the source of blood that we're cleaning and balancing. It's the source of blood that's providing everything that those hepatocytes need to stay alive and in homeostasis. And so then if we look at this other picture here, we're zooming in to where those hepatocytes line the sinusoid. And so the sinusoid is represented by this space here, and you can see those hepatocytes with the microvilli creating the brush border, and then there are those erythrocytes. And so what's separating the hepatocytes from this, the erythrocytes, are those endothelial cells. And then the space is the sinusoid. And so as blood flows through that sinusoid, the oxygen that they need is going to diffuse into those cells, while the carbon dioxide they need to get rid of is going to go into the sinusoid. So it's kind of like this mesh and mixture of arteries and veins together. So the hepatic lobules are pretty easy to identify because they're separated by this connective tissue that we refer to as the stroma. And between the lobules, you'll see this little triad kind of squeezed and pushed off to the side of one lobule and like in between two lobules. And that structure is referred to as the hepatic triad. So the triad has two vessels, an artery and a vein, portal vein to be specific, and then it will have a bile duct. The artery and the vein are going to go from the external or superficial part of the lobule towards the central vein, while the bile duct is actually going to travel in the other direction. And as it does, it's going to collect bile from those liver cells, and it's going to eventually form the bile duct within the hepatic triad. And so those three will always go together, and they're called the hepatic triad. So this image shows 4 different hepatic triads. We can see one here, here, here. And so each hepatic triad will consist of the hepatic artery, which is in red, and then the portal vein in blue, and the bile duct that is in green. And they are all going to be wrapped together in this connective tissue layer that is called the stroma. So our liver has many different cells in it, which is going to give it multiple different functions. The macrophages there are going to allow the liver to function to remove bacteria, and those macrophages are also going to break down red blood cells. They will help to detoxify the blood from drugs, and so will those hepatocytes. Those hepatocytes can actually absorb toxins. They can also store glycogen and make fats in the form of steroids. They can produce proteins. They can store iron and copper. And then these hepatocytes are also going to secrete bile, which is that greenish fluid that is responsible for breaking down fats. If you look at the microscopic anatomy and images here, you can see this is a lobule here. And so this would be another lobule, this would be another lobule. And what's surrounding the lobule is that connective tissue sheath called the stroma. And in the middle is the central vein. Well, pushed off to the side and wedged between two lobules is that hepatic triad, where we have the portal vein that's the largest, and then you have the bile duct and the artery. This is another image of everything labeled or highlighted. So you can see that the liver lobule is highlighted in blue, the central vein here, the stroma, and then we have the triad, and then you got the triad broken down with the portal vein. You have the artery and the artery up here, and then the bile duct here. When we look at these microscopic images, you can identify the hepatocytes because they are the purple or the red. Where you see the white, that is the sinusoids or the spaces, and that is where the blood is actually flowing through, anywhere where you see space. So like the central vein here, blood's flowing through it. And then the hepatic cords, represented in blue here, and the sinusoids, blood is running through those. But in purple, where we see those hepatocytes, those are doing the secretion of substances. And then here we have those hepatocytes on higher magnification. And so wherever you see the stain, that is going to be the cell. And then you can also see those macrophages. Those cells are highlighted here. So our hepatocytes are pretty busy. They're doing a lot of things all at the same time. So, for example, after a meal is consumed, your body is going to break down those nutrients into their monomers or building blocks. So when you eat carbohydrates, the food gets broken down into monosaccharides that we more commonly say glucose. Lipids get broken down into glycerol and fatty acids, where proteins get broken down into amino acids. And nucleic acids get broken down into nucleotides. So these hepatocytes, as the blood is flowing from the portal vein to the central vein, they're going to start grabbing onto those minerals and those monomers and building blocks because we need them to make new cells and to do the things that our body needs. So we're going to grab glucose and amino acids, iron, and other nutrients that we need for metabolism. Or we might not need them right now. So if we don't, we're going to store them. And then, they're also going to be secreting substances into the blood, like clotting factors and angiotensin and lipoproteins. While they're also removing toxins and bile pigments, and they're breaking down hormones. And then between the meals, they are, since they're not absorbing nutrients, they are breaking down glycogen that's stored, and they're releasing that glucose back into the blood. So they're very, very busy cells. Once blood is filtered through those sinusoids, it's going to start to get collected in the center of the lobule in that central vein. And ultimately, the blood is going to flow into the right and the left hepatic veins, and then it will leave the liver at the superior surface of the liver, and it will immediately drain into the inferior vena cava and go to the heart, which is why this filtration is so crucial, because we don't want any toxins or things that can harm us to get to the heart. Then not only will it harm our heart, it can get delivered to anywhere in the body. These hepatocytes are making and secreting bile. And so alongside these hepatocytes, we're going to have these little canals called bile canaliculi that will, the liver cells, the hepatocytes will secrete the bile into. And so this bile will pass into these caniculi, and then it'll eventually pass into a duct of the hepatic triad. Then ultimately, it will lead to the right and the left hepatic ducts, and then eventually to the common hepatic duct, and then into the duodenum of the intestines to start breaking down fatty foods. If that, if it's between meals and if the sphincter is closed to the duodenum, well then the hepatic duct will get filled and bile will go into the cystic duct and then into the gallbladder, where it can then be stored. So some bile is always stored. if you have a gallbladder. But this is a good picture because it shows these bile sinusoids that will, so we start with the canaliculi, and then the bile sinusoids go into the bile duct. And then that bile duct will eventually lead to the left or the right hepatic duct, and then to the common hepatic duct. And then it will travel to the duodenum, and it will meet with the pancreatic duct, where it will form the ampulla of Vater, and it will push out into the duodenum, where it will form that papillae. So if this sphincter is closed, then this common bile duct will get backed up, and the bile can go down the cystic duct, and it can stay in the gallbladder for storage. So near that duodenum, that is where we see the bile duct join the duct of the pancreas, and it's going to make this little swelling. Anytime we see this swelling, it's called an ampulla, and this ampulla of Vater is more commonly now referred to as the hepatopancreatic ampulla. And that makes a little bit more sense, right? It's where the hepatic, the liver, and the pancreas ducts meet. And where it terminates, it is going to terminate on the duodenal papilla on the duodenal wall. And so there is a sphincter that can open and close to allow the bile and the sodium bicarbonate from the pancreas into the duodenum. And that's called the hepatopancreatic sphincter. Some guy named Odie discovered it, so it's referred to as the sphincter of Odie as well. So this will regulate the passage of bile and pancreatic juice into the small intestines. Between meals, the sphincter is closed, and during meals, it is opened. So this is a really good picture that just shows those hepatic ducts, and it shows that hepatopancreatic sphincter, as well as the ampulla and the papillae. Here we can see another diagram, but this is just kind of putting everything all together and in relation for you. So you can see that the esophagus is actually posterior to the liver, and the liver is anterior to the stomach. And then posterior to the stomach, you can see that pancreas, which is that thin leaf-like organ, and then the spleen over here as well. And then you can see the gallbladder on the underside of the liver. Then you can see that cystic duct, as well as the right and left hepatic ducts going to the common bile duct. And then you can even see the pancreatic duct here in the pancreas, where it's going to meet in the hepatopancreatic ampulla right at the head of the pancreas. And then where that papillae is going to be right in the first portion of the duodenum. Here we can see those ducts that are now all highlighted to emphasize where they are in a cadaver. And it also shows that hepatopancreatic ampulla and the papillae. So you can see the duodenum here, and then you can see that papillae sticking out in that location. So the gallbladder we're going to talk about with the liver because they work hand in hand. This is a little small pear-shaped sac that's on the underside of the liver, and it is held to the liver by this in this little groove, but it's attached by the bile duct. And so its function is just for storage. So you can live without your gallbladder because it doesn't actually make the bile, it just stores it. So when you have extra bile that's made, it gets stored here, and that bile, next time you eat a fatty meal, can get released into the cystic duct, where it's going to then meet with the common bile duct of the liver, and it will then travel to the duodenum of the intestines to help break down fats. And so the gallbladder concentrates bile by removing the water. And if we have too much water that's removed, we can have the formation of gallstones. And that will be formed from this crystalline cholesterol salt that is within the bile. Your gallbladder is lined with a highly folded mucosal layer, and so it is a simple columnar epithelium. The head, it typically projects outside of the liver, and then the skinny portion is called the neck, and that's what leads to the cystic duct. Your bile is produced by the liver and it is stored by the gallbladder. And so the contents of this bile is a lot of different things. There are minerals. There is phospholipids, so meaning there's fat. There's cholesterol, another type of fat. There are also bile pigments, so things like bilirubin and bile salts, which create acids, bile acids in liquid. So the principal pigment comes from bilirubin. And bilirubin is from the breakdown of your red blood cells. When it's time for your red blood cells to get broken down, it's going to happen in the liver, and they are made up of hemoglobin. So hemoglobin gets broken down, and one of the substances that's broken down into is bilirubin. And that's going to be what causes the pigments. So this bilirubin is released in the bile, and it will travel to the intestines in with the bile. And so once it reaches here, bacteria are going to play a very big role. And so one of the things that they're going to do is they're going to take that bilirubin, and it's going to break it down into verdirubin, which is like a greenish color, and then it will go to a urobilinogen, and then eventually to a stercobilin, which is what's responsible for the brown color of your feces. And then those bile salts, those bile acids, they will aid in fat digestion and absorption of those fats. If the bile becomes too concentrated with waste, then we're going to start to have gallstones develop. We have a picture of bile. And so you can see it's that greenish color, and the function of it is to break fats down physically. And so we're going to have these large fat molecules that have to get broken down into smaller and smaller and smaller and smaller parts until eventually it is a glycerol and a fatty acid molecule. So the bile will emulsify, meaning it will physically break down and chop those bigger fat particles up into smaller fat particles. And here we have a picture of a full versus an empty gallbladder. So you can see that the empty gallbladder is just kind of this pinkish tissue, right? Because epithelial tissue is, in mucosa, is that light pinkish tissue. But then over here we have a full gallbladder, and it's called robin's egg blue, because when we have that pinkish color of the epithelium plus the green of the bile mixed together, it's going to give that blue color. So the bile will get to the gallbladder by first filling up the common bile duct, and then whatever overflow there is will go into the gallbladder. And a lot of you guys usually have some medical background, and so you know what a cc is. Well, 1 cc is 1 mil. So think about mils in reference to cc's. So the liver secretes about 500 to 1,000 mils of bile a day, which is approximately 500 to 1,000 cc's or I guess it would be like 100 tablespoons or teaspoons, I'm sorry, teaspoons a day. So when it comes to this bile that's secreted, 80% is going to get recycled. It's going to get released into the duodenum or duodenum of the intestines, and it will travel with the food. And once it reaches the ileum of the small intestines, it's going to get reabsorbed, and it will get returned to the liver. Then the hepatocytes, so now it's in the portal vein, then the hepatocytes are going to reabsorb it and then resecrete it. And so that's called the enterohepatic circulation. It's this route of secretion of bile, reabsorption of bile, and then the resecretion of the bile, which happens about two-ish times during digestion. And then the 20% left over is going to be excreted in the feces. And this is the body's only way of getting rid of excess cholesterol. Otherwise, any extra cholesterol is just going to stay circulating in the blood, which can cause plaques and build up and lead to heart attack and stroke. So the liver only has to synthesize or make new bile acids to replace that 20% that is lost in the feces. So some troubles that we can have with the gallbladder are the formation of gallstones. So gallstones are hard masses that are formed either in the gallbladder or within the bile ducts. And these are made up of cholesterol, calcium carbonate, and bilirubin. So here we can see a bile or a gallbladder that's filled with these gallstones. And cholestasis is the formation of these gallstones that is common in obese women over 40. When they have excess cholesterol, it's going to cause this gallstone. And then we can also have those ducts become obstructed. And if we obstruct those ducts because of these gallstones, then we're going to start to see jaundice. And that's going to be this yellowing pigment in the skin. We're also not going to have good fat digestion, and we're going to see an impairment in absorption of vitamins that rely on fat to be absorbed. And then we can also have some infections. And so this image is showing the 3 phases of infection. Over here in the top left, we can see the inflammation beginning. We start to see some redness and inflamed tissue. And then in the bottom here, we start to see areas of bruising and possible necrosis, which is tissue death. And then in the upper right here, we can see that this gallbladder is full of pus and it's broken right open. So we'll continue with the next lesson, and I will talk about the pancreas.